Hello everybody and welcome back to Divulgando Ciencia. My name is Gustavo Herzhuber. In this video, I'm going to talk to you about the variant Omicron. I'm going to tell you what is its origin. Well, let's say the hypothesis of its origin. There are three hypotheses. But first of all, I would like to ask you to please give the video a like, share, subscribe, click the notification bell, and follow me also on Facebook and Instagram. Welcome to this new video. The advent of Omicron is ample evidence that we are not yet done with COVID, and COVID is not yet done with us. To look forward and understand where the virus is heading, we need to understand the origin of Omicron. One thing we can be relatively certain of: where this variant came from, more will come. Omicron is unlike any other variant currently in circulation. The variant carries 50 non-synonymous, 8 synonymous, and 2 non-coding mutations compared to the original Wuhan strain. The three most likely origin stories are that Omicron emerged from an immunocompromised patient, that it emerged from reverse zoonosis, human to animal transmission, followed by animal to human transmission, or that it emerged from treatment of a COVID-19 patient with the mutagenic drug Monopyravir. Here we explore the evidence supporting each hypothesis. An immunocompromised patient. Viral variants are well documented to arise in persistently infected immunocompromised COVID-19 patients treated with antiviral drugs and antibodies. The London patient, the Boston patient, Pittsburgh and Italian patients. In the face of our best weaponry of the time, the virus not only prevailed but became even stronger, learning how to evade our immune defenses. The variations that evolved and emerged from these patients had a wealth of mutations, not just in the spike protein but throughout the entire genome. Omicron carries some mutations identical and others similar to those found in these. Reverse zoonosis. The next hypothesis to consider is that this particular variant evolved from a reverse zoonotic event. Most agreed now that SARS-CoV-2 originated in bats, a reservoir for a diverse array of coronaviruses. The theory with Omicron is that a human strain of the virus jumped back into animals and then re-emerged from that animal to infect humans again. The likelihood that this may have occurred is entirely plausible and may indeed be probable. Since its emergence, this particular coronavirus has infected our entire biosphere, from minks and mice to deer and rats, not to mention our own selves. In each of these animal populations, just like in humans, the virus has evolved and adapted to immune pressure, so far surviving in each population to infect and reinfect. A closer look at the mutations in the Omicron variant and those seen in rats suggests there may be a link. Omicron shares numerous identical and non-identical mutations within the proteins with the wastewater viruses, including 13 amino acid changes in the same position or within one amino acid. Some experts believe, due to these similarities, that if Omicron evolved in an animal host, it is likely to be a rodent. Molnupiravir induced. The final theory, and perhaps the most troubling one, is that Omicron is a result of our own doing throughout the treatment of a COVID-19 patient with the highly mutagenic antiviral drug Molnupiravir. Molnupiravir works by introducing errors in the virus's genetic code. When enough errors are introduced, virus replication slows and the patient clears the virus. Under less than ideal conditions, that is, when the full dose of Molnupiravir is not taken over the full period of 5 days, for example, the drug could lead to the creation of highly mutated but viable strains of SARS-CoV-2. Even under ideal conditions, patients treated with Molnupiravir produce viable virus a few days into their course of treatment. The extent of the mutations which appear due to Molnupiravir are significant. In the FDA analysis of Merck's clinical trial results, The authors note that patients who received Molnupiravir showed more viral variation than those who did not, including amino acid substitutions, deletions, or insertions in the spike gene, and amino acid changes were scattered throughout the coding sequence. A total of 72 emergent spike substitutions or changes was detected among 38 Molnupiravir treated patients. In South Africa, where Omicron was first detected, Molnupiravir has been taken in both ideal and non-ideal conditions. 4 different South African locations were used in Merck's clinical trial of Molnupiravir, which began in October 2020. The drug was given to patients at what we now know to be the optimal dosage, but also at lower doses to test the drug's efficacy in smaller amounts. There is by no means a foolproof connection between Molnupiravir and Omicron, but Molnupiravir is known to induce a preponderance of 2 types of mutations, cytosine to uridine or C2U and guanosine to adenosine or G2A. If you look at the difference in the Omicron genome and the original Wuhan variant, these C2U and G2A mutations comprise the majority of differences, with C2U mutations more prevalent than G2A. The same has been observed for Molnupiravir-induced mutations in other coronaviruses. There is still much more study to be done before we will know with any degree of certainty which of these 3 scenarios led to Omicron's evolution. But we know enough today to make a few assumptions and assertions. While Omicron may be bad, future variants could be far worse. The United States and many other countries are still far behind where they should be when it comes to testing and genomic sequencing of viruses in circulation among humans and in other populations. Until we do better on this front, until we understand that the virus has already achieved and how it succeeded, we will never have a full understanding of what more this virus can do and what is ahead in this pandemic. Omicron tells us that now, more than ever, we need to institute a systematic multimodal approach to COVID control, including public health measures, vaccines, therapeutic and prophylactic drugs, and collaborative global engagement. As you could see on this video, there are 3 main hypotheses about the origin of the Omicron variant. The first one is the one of the immunodeficient patient, which is the hypothesis also for the previous variants. The second hypothesis is the one of the inverse zoonosis, and the third one is the one about the drug Molnupiravir from Merck. But what about a 4th hypothesis where you have a combination of 2? For example, an immunosuppressed patient that took Molnupiravir. Please let me know in the comments below what is for you the best hypothesis. How do you think Omicron origined? If you like the video, please give it a like, share, subscribe, and also follow me on Facebook and Instagram. This would help me a lot. Thank you very much and see you next time.